hello to all the students of BPC. This is your Prasanna ma'am from NEET World. We here in the NEET World are providing CUET entrance exam coaching where you will get complete analysis on all the previous year questions and complete syllabus in detail explanation with mock tests. So, for the students who are going to write in the 2025, this session is really useful for you where I am going to deal the biology question paper which covers these chapters, that is sexual reproduction in flowering plants, principles of inheritance and variation, molecular basis of inheritance, microbes in human welfare, biotechnology and its applications. From these chapters, if we consider, these are the set of questions beta. So yes. So if we look into the first question, who performed experiments on Visia faba to prove that DNA replicates semi-conservatively? For semi-conservative mode of DNA replication, experimental proof for prokaryotes is given by Michelson and Starr. It is given by Michelson and Starr. Okay? By taking the model organism E. coli. And if you take in eukaryotes, it is Taylor and his colleagues who has proved that DNA replicates semi-conservatively by taking the model organism Visia faba. Do remember that beta in both prokaryotes and eukaryotes, it is very important. And second question if you take, this is from PCR, polymerase chain reaction. As such, we got 2 to 3 questions from that concept. A thermostable DNA polymerase is isolated from, that thermostable DNA polymerase is isolated from Thermus aquaticus. It is isolated from Thermus aquaticus, okay, which has the enzyme that is Taq polymerase, which is thermostable, okay, that is extracted from Thermus aquaticus. And next if you see third question, that is from ELISA, okay, enzyme linked immunosorbent assay is based on the principle of specially that is antigen antibody interaction. This is mainly based on antigen antibody interaction. And next question if you look into, which of the following represents a test cross in which half of the offspring is heterozygous and next half would be homozygous? Test cross, we all know, right? What is test cross? Crossing dominant phenotype with recessive parent, dominant phenotype with recessive parent is called as test cross. So test cross is used to identify the genotype of the organism, okay, test cross is used to identify genotype of the organism. So here if you look into dominant phenotype, that is heterozygous crossed with recessive homozygous, this will be the cross. So what will be the option over here? Capital T, small t, small t, small t. Option 2 will be the correct answer. And test cross is used to identify the genotype of the organism, identify the genotype of the F1 generation specifically. And next question if you take, this is from cell cycle and cell division. Next question is from cell cycle and cell division, wherein in which phase of cell cycle replication of DNA takes place? Replication of the DNA takes place in S phase of the cell cycle. Okay, replication of the DNA takes place in which phase? S phase of the cell cycle. That is option 3. Next if you take, in Hershey and Chase experiment, some viruses grew on the media that contain. Hershey and Chase gave unequivocal proof that DNA is the genetic material, right? So here Hershey and Chase experiment has proved this, wherein the bacteriophage, okay, at the tails it has lysozyme enzyme. This is on the bacterial cell wall. This will degrade the bacterial cell wall and it will inject this genetic material directly into the bacterial cell. So this Hershey and Chase experiment has proved proved that DNA is the genetic material, wherein they added 2 radioactive isotopes, one to the protein layer, that is protein sheet of the bacteriophage, that is S35. Okay, that radioactive isotope is S35 radioactive isotope, and the genetic material has P32 radioactive isotope. Based on that, 3 steps: infection, blending and centrifugation. After centrifugation, they have noticed that in the supernatant only this protein sheath is there which has S35, but P32 isotope is not detected in the supernatant. It is injected into the genetic material of the bacteria. That is why they said that DNA is the genetic material. Fine. So here which option we have to choose, Nana? What are the 2 radioactive isotopes? P32 and S35. That is option 1. And next question, if you look into, which of the following is not a process of DNA recombinant technology? What is not a process of DNA recombinant technology? In DNA recombinant technology, we will be using the very first step is isolation of the DNA, and by restriction enzymes, both by restriction exonucleases and by restriction endonucleases, DNA fragments are formed, and they are poured into the agarose gel, wherein after that lastly culturing of the host cells in the medium at the large scale is done. But nowhere in the DNA recombinant technology we are using RNA interference. No way we are using RNAi. This is not used. So here the option will be option 2. And next question, if you see, ex situ conservation. Ex situ conservation includes what? All the biosphere reserves, national parks, wildlife sanctuaries, these are all comes under in situ conservation, beta. Whereas seed banks, pollen banks, all these comes under ex situ conservation, where we are preserving those seeds outside their environment. So this is option 4. In velvet grass seed, the cotyledon is called as, grass seed is what? Monocot. Okay, monocot seed has only one cotyledon, that is papery cotyledon scutellum. What is that, Nana? Scutellum. This coleoptile is the outer sheet of the plumule. Coleorhiza is the outer sheet of the radicle. Testa is the outer seed coat of the seed, and tegmen is the inner seed coat. Next, if you see, which of the following is the recessive trait for garden pea plant? Recessive trait, if you see round seed, that is dominant, tall plant is dominant, violet flower is dominant. What is recessive? Constricted pod is recessive. Constricted pod is recessive. Inflattened pod is dominant. Next, if you see, nucleosome is having what? Negatively charged DNA wrapping around positively charged histone octamer. So if you see, what are the histone proteins involved, beta? H2A, H2B, H3 and H4. 2 pairs of this proteins, 2 sets of this proteins, that is histone octamer. Histone octamer. Around this histone octamer, DNA which is negatively charged is wrapped around this positively charged histone octamer. Thereby, DNA condensation happens. So here what we are looking into, ma, nucleosome is negatively charged DNA wrapped around positively charged histone octamer. That is option 2. Next, transforming principle was given by Frederick Griffith. By conducting experiments on Streptococcus pneumoniae, and he has discovered the transformation principle. Next, if you see, which disorder is caused by substitution of glutamic acid by valine at 6th position of beta globin chain in the hemoglobin? That is point mutation. Point mutation, wherein 6th position of glutamic acid is changed by valine, okay, just by changing one nitrogen base. So that is with sickle cell anemia. Okay, so here the answer is sickle cell anemia, which is autosomal recessive disorder, and that is point mutation. Next, arrange the following steps of DNA fingerprinting in the proper sequence. So here the very first step of DNA fingerprinting might be, recombination might be, the very first step that we are doing is isolation of DNA. And here by just looking into this, the option is very obvious beta. So here isolation of DNA, after that digestion of DNA by restriction endonucleases, second thing, and then separation of the DNA fragments by electrophoresis. After that blotting of separated DNA fragments on nylon, and then the last is hybridization with variable number tandem repeats probe, hybridization with this, that is option 2. And next, if you see streptokinase, this is from microbes in human welfare. The enzyme is used to remove the clots from the blood vessels. Okay, this enzyme is used to remove the clots from the blood vessels. Next, again here, okay, incorrect statement we have to find out. Chromosome 1 has most genes and chromosome Y has fewest. This is true only. Chromosome 1 has more and chromosome Y is fewest. Chromosome 21 has most genes and chromosome Y has fewest. Wrong. Chromosome 21 is just of 1.5 to 2% of the total DNA in the cell, whereas Y chromosome is little more than the 21st chromosome. So this is wrong. Less than 2% of genome codes for proteins. Yes, true. The functions of over 50% of discovered genes are unknown. Exactly correct. So here the wrong statement is option 2 ma. This concepts especially from molecular basis, you will at least get 2 to 3 questions from these areas beta, that is Human Genome Project and DNA fingerprinting. Please look into it once. So next, if you see again from polymerase chain reaction, second question in the same paper, wherein which of the following is not a step for PCR? Not a step for PCR he is asking. That is the PCR, the very first step is denaturation. First step is denaturation. Second step is annealing and third step is extension. Denaturation, annealing and extension. These are the 3 steps of polymerase chain reaction. The step which is unnoticed for PCR is downstream processing. Next, if we look into this table, wherein I gene codes for repressor, option 4. A is for trans-acetylase, Y for permease and Z beta galactosidase. So once if we quickly recall the Lacoperon model, that is gene expression model, Lacoperon, wherein first you will have promoter followed by I gene, again followed by promoter and then operator. After that you have 3 structural genes, that is Z, Y and A. Lastly it is terminator. So this is from 5' to 3' end. To promoter region or always RNA polymerase will bind. Do remember beta, to the promoter region always what will bind? RNA polymerase will bind, and I gene will release repressor. To this repressor lactose will come and bind. To the promoter again RNA polymerase will bind. To the operator region, if lactose is not in the medium, repressor will bind and it will inhibit the process. And most importantly we have to remember is Z gene codes for beta galactosidase, Y gene codes for permease and A codes for trans-acetylase. So here if we look into the options, A is 4, B is 3, yes, C is 1 and D is 2. Option 2 is the correct answer. Next, if you look into amino acid is attached to which of the tRNA, to which site of the tRNA. So if we take tRNA, it has inverted L shape and clover leaf shape. Inverted L shape is the tertiary structure and clover leaf shape is the secondary structure. This is the 3' and this is the 5'. Close to the 3' you have T psi U loop, close to the 5' we have DHU loop. So at the 3' amino acid which is charged, amino acid which is activated along with ATP, this amino acid binds with 3', this will charge the tRNA and makes the tRNA ready for translation process. So the very first step of translation is amino acylation of tRNA, that is amino acid adding at the 3' end. So here the option is 2. Which of the following plays significant role in our stomach in checking the disease causing microorganisms? That is lactobacillus. Okay, lactobacillus in the stomach, it will increase the microvilli absorption. It will increase the microvilli absorption and it is highly useful for the digestion as well. That is lactobacillus. That is why it is most noted to eat curd after the lunch. Okay, lactobacillus is very much useful for the healthy gut. Next, if you look into the statement based questions, whiskey, brandy, rum are produced without distillation of the fermented broth. Wrong. All these are distillated. They are distillated. They are not without. They are produced without, not without distillation. They are produced through distillation. Without distillation, it is wine and beer that is produced. Next, Saccharomyces cerevisiae is doctor's yeast. Wrong. Saccharomyces cerevisiae is called as baker's yeast or brewer's yeast, not doctor's yeast. This is also wrong. So here two statements are wrong, but statement 1 is false and statement 2 is also false. Both the statements are wrong, that is option 2. Given below is a DNA coding sequence. Its complementary strand would be, okay, what is the complementary strand for this? 5' to 3', this is there. 3' to 5', which sequence will be complementary? If you see 3', that is to the coding strand, the complementary sequence is template strand of DNA. So here if you see, Nana, G is CATAATGC. Okay. So what is the correct answer here? Of course, option 2. You have to look into whether it is, he is asking you the complementary sequence, then you have to code for this. If the question is about, same as that of coding strand of same as that of coding sequence, or he is asking about mRNA, then the sequence will be same except at T position you have to write U. Okay, if the question is about mRNA, at just T place you have to change U. That's it. Everything else will be same. Next, if you look into which of the following statement is not correct for restriction enzymes? Which of the following statement is not correct for restriction enzymes? Restriction exonucleases remove nucleotide from the ends. True. Restriction endonucleases make cuts at specific positions within the DNA. That is also true. Ligases join sticky ends of DNA. Wrong. Ligases will join the DNA fragments, not the sticky ends. Sticky ends are joined by what? Restriction enzymes are involved in that, joining the sticky ends, not ligases. They will just join the DNA fragments. So here that is option 3, which is wrong. Fourth statement, if you see, first restriction endonucleases is HindII. This is also true. Next question, if you see, which of the following is not associated with megasporangium? Okay, funicle. Okay, funicle is the stalk of the ovule. This is associated. Integument, which forms the outer layers of the ovule. That is also true. Micropyle, which is the pore of the ovule. Okay, which forms a pore in the ovule. That is also true. So here the right answer is generative cell, because generative cell is nowhere associated with megasporangium. Generative cell is nowhere associated with megasporangium. Okay, generative cell or vegetative cell are with respect to pollen grain. Next, in diploid organisms, which of the following undergoes meiosis? If you clearly see, Nana, vegetative cell is haploid, pollen grain is haploid, synergids are haploid because they are in embryo sac. Okay, and sporogenous cell is, or sporogenous tissue is diploid, which has microspore mother cell, and that undergoes meiosis. That is option 2. Next question, if you see, which of the following controls the growth of bollworms? Here he didn't mention clearly stating either cotton ball worms or cone borers. So just he said ball worms. So in that case we have to choose both cry 1 AC and cry 2 AB. And here if you see the last set of questions from this chapters, wherein when one parent has A blood group and other parent has O blood group, then their child can be of what blood type? Okay, then their child can be of what blood type? One is of A blood group, another is of O blood group. Okay, then their child is of what blood type? They are asking. Definitely if you cross, all are of A blood group only, no? Okay, all are of A blood group only. All are of A blood group only, that is only one, only A, that is option 1. Next, the size of variable number tandem repeats varies from, the size of variable number tandem repeats, they are of small repetitive DNA sequence, they vary from 0.2 to 15 kb. They vary from 0.2 to 15 kb. They are of small repetitive DNA strands. So with this we have to discuss this paper beta. There is one more set of CUET paper. We are going to discuss it on Monday. So stay tuned for more such updates, and I would like to end this session by clearly saying you that here in the NEET World, which is in Narayanaguda, Hyderabad, both offline and online batch is commencing from May 20th. Okay, May 20th, 2025, and second batch is from June 1st, 2025. So whoever are interested can ping us up, can call us up by before number. Okay, thank you.